I'm Viet Nguyen. I'm a neurologist, a clinical neurophysiologist, and running clinical development at Actio Biosciences. I've been working in genetic epilepsies and neurodevelopmental diseases for a few years, but I joined Actio this year. I appreciate the opportunity from the foundation to present to this roundtable and introduce the KCNT1 program to the group. As most people are aware, Ali just presented her own version of the slide. The KCNT1 related epilepsy is a severe monogenic epilepsy syndrome, so not just seizures. It lacks effective treatment options. There's intractable seizures, usually onset during infancy, with developmental delays and regressions, early mortality in many cases. We think the epidemiological data is varied and sparse, but we believe there are at least 2,500 patients in the US and at least that many, probably more worldwide. Hopefully, we'll discover more of these as genetic testing becomes more commonplace in the pediatric epilepsy space. The seizure burden is high, and in our analysis of a portion of the citizen electronic medical record cohort set, we found that patients are having about 20 seizures on average per day around the age of 4 years old. So quite a bit of seizure burden for a developing brain. As far as we understand, this syndrome is driven by pathogenic gain-of-function variants of the KCNT1 gene. The gene codes for potassium ion channel. It's expressed in the central nervous system, especially in interneurons. I think that's an important part of the story. Our therapeutic hypothesis is that if we can characterize the molecular function of these mutant channels, and then design an inhibitor that will shut down all of them, each of the mutations, then perhaps we can make a dent in these seizures and normalize neurodevelopment. So that's what the translational team at Actio has done. They created in vitro models of all of the known pathogenic and likely pathogenic variants, 73 variants in total at the time of that analysis. About 67 of them we were able to create functional models of, and we found that indeed, most of them exhibited gain-of-function activity, as you would suspect. Then we worked with our stellar chemistry team to design a series of molecules that would shut down that gain-of-function activity. You can see here in the blue curve. We've chosen the best of those molecules, ABS-1230, to advance into human studies. And by best, I mean it worked for the mutations, as in all of them, and it inhibited their gain of function strongly and consistently, and it did so at very low concentrations. So this is where our program started, and this is where we are at right now with the candidate we'd like to advance, ABS-1230, in terms of their potency in shutting down gain of function. So this leads to an almost unheard of 0.3 nanomolar human potency, without messing around with at least 500 other off-targets that we've tested, and with a 20-hour half-life. This means we predict that ABS-1230 will be able to inhibit the mutant channel, whatever that mutation might be, only that channel, not mess around with anything else, when given at low doses, once a day, orally or via feeding tube. Preclinical studies are completing now, and we're busy preparing our regulatory interactions that will allow us to enter clinical trials. The trials will be in KCNT1 related epilepsy first, although we are also looking at other interneuronopathies that this might be helpful for. Those interneuronopathies lead to other forms of developmental epileptic encephalopathies. The trials will look at all the things that we feel are important for patients: the seizure burden, the neurodevelopment, quality of life, the burden on caregivers, and EEG. Our preclinical proof of concept used the R409Q homozygous mouse model, and it demonstrated significant resolution of the seizures on drug and return of those seizures after you washed the drug out, meaning that it was the drug that was doing it. And then the corresponding changes you would expect to see on EEG, resolution of seizures and return of those seizures. This R409Q mouse model is that mutation is kind of the equivalent of the human R428Q variant. So we plan to start a healthy volunteer study this summer for the small molecule, with a first in-patient study in early 2026, and our pivotal study after that, with all of the completed patients eligible to join our open label extension and stay on the drug for long-term outcomes and safety. Thanks for allowing me to present to the group.